I already have my presentation up. So this the panel debate is based on the consensus statement of ISAP on the definition and scope of postbiotics, of which you see the publication on the left, and a reaction to that consensus statement by some other authors, of which you see the publication on the right. I think they kept the discussion nicely. They didn't fight. They didn't throw any dirty words or whatever, but they do disagree, it seems. So we decided to invite some of these authors here and give them a brief period of time to explain their ideas. And then we'll actually have a debate we're going to have to move around the podium here a little bit with tables here and see, if we can really come to a real consensus on what postbiotics are. So the first speaker in this session is Gabriel Vinderola from Argentina. He's a principal researcher at the National Scientific and Technical Research Council, or CONICET, as you can see, and associate professor at the Food Engineering Department of the Faculty of Chemical Engineering from the National University of Littoral. And he serves as a member of the board of ISAP from 2020 onwards. After him, we have Professor Lorenzo Morelli, an old friend whom I've known for more than 30 years now. Unfortunately, he couldn't be with us here live, so he is going to join us on Zoom, which is also why we have a camera in the middle of the room, so he can actually follow the debate as well. And he's professor of food microbiology and head of the Department of Sustainable Food Processes at the Faculty of Agriculture, Food and Environmental Sciences at the Università Cattolica in Italy. But first, Gabi, floor is yours for a brief introduction on the concept of postbiotics.
Thank you, Kim. I will try to be as brief as possible. Thanks for this invitation. First, I would like to say very briefly that I started studying lactic acid bacteria in November 1995. And by that time, Professor Lorenzo Morelli was really relevant. I learned a lot from him. And I'm the 5th generation of Italian immigrants. So Italy, Lorenzo were really always, guiding light. So participating in a panel debate with him is an honor and it's a challenge. So I hope to make my best. Okay, ISAP, I think you know ISAP. It's a non-profit organization. It's committed with advanced science on probiotics, prebiotics, and related substances. And one of the activities has been proposing a consensus definition. ISAP is not a regulatory agency, and as Colin Hill always said, ISAP is not the term police. But this definition, they create quite attention, and in total, they had more than 500,000 accesses: the probiotics, prebiotics, symbiotics, fermented foods, and postbiotic. One of the characteristics is that these definitions are made by the group of people. Some of them belong to ISAP, some of them are invited. They are outside ISAP, and people from different countries, different perspectives, different opinions. And sometimes it's really hard to have an agreement within the panel that proposes the definition. So we saw in Antwerp, before the pandemic, an opportunity to say something about non-viable microbes and the metabolite. This is a review from Seppo and from Arthur Owihan that 25 years ago, they say, okay, non-viable microbes, they are metabolites, they can induce a health benefits. And if you take a look in the literature, this phenomenon was called out with many, many different terms. As you can see there, heat inactivated, probiotic, heat indolized, paraprobiotic, inactivated, self-fragment, side-lysates, non-viable, ghost probiotics. So imagine how confusing can be this for consumer, for physician, for regulatory agencies, that we are using many, many different terms to refer almost to the same thing. So we say, okay, maybe we have an opportunity to collide all these terms in a single term. And at the same time, in the market, there were already products containing non-viable microbes with or without metabolites. You can see a mixture of lactobacillus for diarrhea, a fermented infant formula, heat inactivated Aspergillus oryzae for animal nutrition. You can see the famous spirulina that may fit in the or ambient yogurt that doesn't need refrigeration, or the famous bacterial lysates that come from pathogens, but that can be used to boost immunity. So there was a group of these non-viable microbes and metabolites. Of course, there was the term paraprobiotics was already published, but we saw a couple of limitations. One of the limitations was that the paraprobiotic had to come from a probiotic. So you have there a first challenge. You have a double work. You have to prove it's a probiotic, and then you have to prove that when it's dead, still have an effect. And if you go and read the paper, it says ghost probiotic, and ghost probiotic is defined as the shell, as the capsule, the membrane, the cell wall, not the cytoplasm. So we say, okay, paraprobiotics maybe is a term that is really limited because it's related to something, to a shell that comes from a probiotic. How do I go back? In fact, by that time, there were 6 definitions of postbiotic. If you want to take a look, you may find 6 definitions. Some of them, they share some things, but of course, there was no agreement between all of them. And if you consider that definitions can change over time, if you go and take a look to the definition of probiotics from 1965, it has nothing to do with the definitions we use today. So concepts can evolve to represent science, and that's nothing wrong. I think how science is constructed. So definitions can change, and not necessarily the first definition is the best one or the one that best represents the science. So if you go and take a look to the first definition, the 2013, we also found some limitations. For example, it says that a postbiotic are metabolites produced by a probiotic. So it requires a probiotic as a progenitor, creating the untenable situation in which a metabolite produced by the established probiotic is considered a postbiotic, but if we had the same metabolite produced by a strain that is not a probiotic, you cannot call it the same. So imagine lactic acid. If it comes from a probiotic, it's a postbiotic, but if it comes from Bacillus cereus, you cannot call it a postbiotic. Then in the definition, it's not clear if you have to administer the postbiotic or if you have to administer the probiotic and expect to produce the postbiotic in situ. And if you keep reading, for example, it will say molecule of factor produced and secreted by the bacteria in the gut lumen. So you are leaving out yeast, mold, you are leaving out skin, vagina, respiratory tract. So it was a definition that was, meant for the gut, for bacteria, leaving out many things. And at the end, the definition then does not state clearly a health benefit. It just says a benefit. So what is a benefit? Maybe it's an economical benefit for the one that produces. So I think we have to be really clear that we are dealing with health, and we have to say that we are trying to produce a health benefits. Why we choose the term postbiotic? Because all the other terms ended with probiotics, and we want to be independent from the progenitor strain. Maybe we can start with a strain that does not fit the definition of a probiotic. Cell fragment and cell lysate do not represent everything, because maybe we have a whole cell, not a fragment. And the term postbiotic, post means after, not from. So from the cloud of words that were used, we felt that postbiotic best represented what we wanted to say. So The definition took more than almost 2 years from the idea, from the discussion until it was published. You know, well, the definition you don't know, postbiotic is a preparation, and preparation we use because the technological process can have an influence. It's not the same to terminate life by heat, by radiation, by high pressure. So the technological process can have an impact. Inanimate microorganisms and or their components that confer a health benefits. The term inanimate, it's correct in English. We had 4 native English speakers. Inanimate means that there is no biological activity, there is no, it cannot grow. Maybe it sounds strange, weird, but it's like that. Science advanced, new words appear, and the people that native English speaker, they say that inanimate was completely right. And we had people from the States and people from London. So, okay, I'm from Argentina, I cannot contribute much. And of course, we have to ask for a health benefit. If not, we are dealing with what? How can you see a postbiotic? A postbiotic can be a single culture, can be a combination of culture, can be a cell with the metabolites all together. It can be a fragment and a fragment with metabolites. So it's really comprehensive, and many cases can be included there. But if you have a postbiotic preparation, I mean you have your microbes, you have your cell fragment, you have your molecules, but if you start purifying, you start removing cells, removing fragments, removing metabolites, and at the end, you end up with a single protein. That's no longer a postbiotic. That has a chemical name. That is a bacteriocin, that is a protein, that is whatever you want. You don't need a new name for something that has already a chemical name. Postbiotic is something, try to capture the complexity of something that can produce a health benefits. I think this, we think that this definition promotes innovation. Maybe you heard about bacterial vesicles, these bacteria that can release part of their cell wall, and that cell wall, it's a fragment of the cell that can induce health benefits. And if you think in Akkermansia, for example, Akkermansia was first approved in the pasteurized form. So it was first a postbiotic and not a probiotic, although we know it can be. So some key points from the consensus paper, that post means after. And we think that the importance of words have an importance. It's not mandatory that a postbiotic be derived from a probiotic. You would have a double challenge, and people who does clinical trial know how difficult it is to make one clinical trial. If you have the condition that a postbiotic must come from a probiotic, you have a double work. And for example, you can even use pathogens that were inactivated. They are no longer pathogen, but still can confer a health benefit, and think in bacterial lysates to boost immunity. And of course, a probiotic that gradually loses viability during storage does not become a postbiotic. You have to make the clinical trial with your microbes dead. And some concluding remarks, some take-home messages. Remember that 6 definitions of postbiotic had been proposed between 2017 and 2021. Some definitions require metabolites be produced by a probiotic bacteria in the gut, leaving out identical metabolites produced by a strain that does not fit the definition of probiotic, leaving out yeast, mold, leaving out other mucosa like the skin, the vagina, the respiratory tract. Requiring that the postbiotic be derived from a probiotic, we think is an unnecessary burden for product development. And we believe that ISAP definition is a clear one, forward-looking definition with rigor, purpose, and it's wide enough to promote innovation. And finally, as we always say, time will tell. ISAP is not a regulatory agency, it's not the term police. Which definition will be pushed forward by scientists, academia, the industry, health professional, consumers, and finally, a regulatory agency. Once the regulatory agency adopts one definition, that definition will be valid for that place, and maybe the country next to you will adopt another definition. So very humbly, ISAP proposes this definition, and if you go in the paper, you can see the details, and you can choose. Thank you very much.
Thank you very much, Gabriel, for this very nice position on the definition of postbiotics. Now, I'm looking at the technical support. Do we have Lorenzo online? We have Lorenzo. Hi, how are you?
How are you? Nice to be there, even if in a digital way.
Good. We can hear you well. So I'd like to ask you to give your point of view, Lorenzo. Please go ahead.
First of all, I have to say that this is not a quarreling situation. It's really a discussion about among scientists, just to try to improve the landscape of the postbiotic area. If I may have my presentation, I think I have. Okay. This is the title, just to be, let's say, in a light way, to address in a light way and to copy Oscar Wilde, the relevant things of to be, let's say, solid in definition. In the next one, I am trying just to simply summarize the situation. We have a lot of definition. I totally agree with Gabriel. We have probiotics, symbiotics, let's say, there are too many, but to produce a definition. And I have spent 13 years of my life in a commission of the Italian Ministry of Health. And just to remind you that Italy was the only one after the failure of the EFSA approval for health claims probiotic to defend the term probiotic. And in Italy, is allowed, has been ever allowed, according to some rules. So I have really learned a lot by this experience. And for the regulatory bodies, and we as scientists, we have to take care about the regulatory body's position. We have to establish methods. We have to provide information on how to check, how to control a product, if this product is accomplishing or not to a definition. Otherwise, we can have problems. Just remember the failure of the health claim situation in Europe. The next one, please. And here I really represent the two main documents that in between FAO and WHO we have produced about the definition of probiotics. Why? Because in the next slide, you can see, following the arrow, a very important things. One of the major worries of this definition is to provide something about how to check live microorganisms. That was the base of this definition. And if we go ahead in the next slide, I'm trying to really summarize this to have more space for the definition. I think we have to keep in mind, together with the term definition, the terms qualification. Qualification are really, really relevant to define something. And in the case of probiotics, we have some qualification. For instance, the correct identification of the probiotics. Just think about a non-correct taxonomy. Now we are living the nightmare of the new genera stemming from the genus Lactobacillus, but you know, we have to be extremely precise. And in the next one, I will present to you something arriving from the Italian Ministry of Health, and it's Ministero della Salute, and they say, definitely, we have to identify this bacteria, also because the regulatory bodies, and this is one of the first points really interesting for the future discussion, I think, they belong to the food area, in Europe to the QPS, in the state to the GRAS. And we have also to make a distinction. Are we dealing with products that have to be classified as drug. In this case, the use of pathogenic bacteria is not a problem, but you have to face phase 1, phase 2, phase 3 approval as a drug. But if we are in the food supplement and food, and remember that food supplements are covered by the food legislation, there is no special legislation for food supplement. Is it feasible to think about the use of non-food related bacteria? The example of Akkermansia is clear. Akkermansia, pasteurized Akkermansia was approved as a novel food, but Akkermansia has not received the QPS status. So, for instance, it can be not used in yogurt for the moment. So I think it's we have to keep in mind the distinction in between food, novel food, food supplement, and drug. And from the scientific point of view, in the next slide, I have tried to collect example of something that is worrying me. This is a really teeny selection of a huge number of marketing advertisement about the presence of probiotic in products in which viability is still to be proven. Okay, we can use spores, but are we sure the spores have a beneficial effect? And what about beneficial effect or health effect? Because FAO and WHO define probiotics for health benefit. From the regulatory point of view, may we still use the word probiotic for different beneficial effect, detergents effect, sanitization effect? And in the next slide, I will show you a very tricky situation. Have a look to the circle. This is the FAO WHO documents. And on the right, you have the table, the scheme, how to arrive to define a bacteria strain as a probiotic. As a probiotic food, as it is written in the central square, not a drug. And there is an arrow missing in between the square containing the words probiotic food and the square containing the phase 3 efficacy, because the phase 3 is related to the drug use. So the missing arrows is by purpose. The working group in FAO, the WHO, and I was a member of this working group, has deliberately missed to use the arrow and to connect the probiotic food definition to the phase 3 that is related to a drug. So just to really highlight what is really to be taken care for a definition. In the next slide, just this was a survey about the qualification of a definition, and I, by purpose, stick to the definition of probiotics just to end with the postbiotics. And it was conducted years ago by the National Institute of Health that in Italy has a regulatory power. And it was a nightmare at that time about the lack of adequate amounts in a lot of products that, I don't know if you can read the abstract there, but the products can be sampled directly in the production plant, just to avoid any complaint about, okay, the shelf life, the shops have not maintained in a correct condition and so on. Since that time, and I was the head of this kind of research. And since that time in Italy, the situation has improved a lot. And in the next one, we can see another qualification. We have to be sure for probiotic, they are able to proliferate into the gut. And also this was an evaluation about the factors affecting the survival or persistence of beneficial bacteria in healthy adults. So even a very a solid definition, as the definition of probiotic is, needs a lot of qualification and provide a lot of measurement ways. In the next slide, we will enter in the postbiotic. And the postbiotic are something defined as also microbiotic, tyndalized probiotic. Tyndalized probiotic is a nonsense because probiotics are viable and tyndalization is a heat treatment. Symbiotics, chembiotics, and so on. And you can see, and I totally agree with Gabriel, the explosion of scientific terms. I think that we as scientists have to be a little bit more prudent in providing new words for our research. And in the next slide, we will see the first use that has been done by my friend Maria Rescigno in postbiotic. And her definition is clearly something related to metabolites. We can question this, we cannot agree, but there is a strong point in this case with this definition. Just try to think as a regulatory people. We have something measurable, produced by something that is already accepted in the food area according to the long history of safe use. And the long history of safe use is of paramount relevance to accept something in the food area. Otherwise, you have to go to the novel food part. That is a totally different one. And in the next one, you have seen, we can skip, let's say, because Gabriel has in an excellent way showed the position of ISAP. But just think about a regulatory people and go to the next slide. What are you going to measure to be sure about the efficacy of your products? The biomass. This is a freeze-dried preparation of a probiotic strain at left at the beginning, at right after 3 months. It is clear that there is a kind of deterioration of the cell mass. So if you don't know which are the metabolites, the cell wall components, the cell components that exert the health beneficial effect, how can you measure? In which way can you establish the shelf life of your product? The shelf life for a probiotics is easy, are the viable cells. And then you can see in the next slide, even more in a greater magnification, what I mean, what is my worry. Okay, and this is the reason why in a group of scientists have published in the next slide a letter in which we have not arguing, but we say to be simple, simplification could be acceptable, but at a certain limit. We have to be precise more than simple. And in the next slide, we have also, this is one of my worry, we have also to think about the consumer, not only to the regulatory people. Consumers are really under an explosion of marketing advertisement. And we have to be prudent. We have to take care of the consumer. In my last slide, I think that we have to do an effort among all we scientists to provide measurable indication using for the regulatory bodies and also for research to improve the product. How can you ferment? How can you stabilize this product to protect the consumer? And really, and I share with, to encourage innovation, but innovation mediated by the sound research. Thanks a lot for your attention.
Thank you very much, Lorenzo. Please stick around. We're going to change the podium a little bit here. And if I may have my presentation back to introduce the other two people that are going to join us in the panel. Which are Seppo Salminen, professor and director of the Functional Foods Forum at the Faculty of Medicine at the University of Turku in Finland. And he was the president of ISAP from 2018 to 2021. And then also Guus Roeselers, who is an old colleague from me at TNO. When we both used to work there. TNO is the Dutch Organization for Applied Scientific Research, but now Guus is a senior group leader, gut microbiology and specialized nutrition at Danone Nutrition Research. So please guys, join me on stage. I'm sorry, this is an all-male panel. This was not intentional, and I didn't think about that before, but next time I'll promise I'll change that. So we heard maybe not entirely different opinions. I think I also heard Lorenzo say that he agreed with Gabriel on certain of these aspects. But I think that if we would do a word cloud on Lorenzo's talk, I think the major word that would jump out would be measurable, right? And then perhaps the second one would be regulatory. So maybe Gabi, can you, or Seppo, or both of you, can you respond to that at first instance?
Yes, I totally agree with Lorenzo that measuring is challenging, but I think it's not impossible. And I think that the same challenges happen with probiotics, that we measure CFU, but at the end of the shelf life, if you make flow cytometry, you will be surprised to find maybe much more dead cells than viable cells. And in the industry, when you produce the biomass of a probiotic, you don't wash cells as we do in the laboratory. So in the industry, you will centrifuge, you will add the protectant, and you will freeze dry. So your probiotic product is full of viable cells, dead cells that we never measure, and that can be really high, and metabolites. And we only pay attention to CFU. So there are only challenges and limitations in how we measure probiotics. And for postbiotic, I think Seppo showed me this morning the approval of Akkermansia, and they count cells, total cells, and we are able to count cells by flow cytometry. So I think we are not that, it's not that different to monitor what is there. We have total cells by flow cytometry, metabolites, but we have metabolites in both. So I think it's challenging, but I think that is something for product developers.
Seppo, you want to add to that?
I could add on agreeing with Lorenzo that we need consensus, especially for the regulatory authorities. Not only the regulatory, of course, for the consumer information as well, but we have a fine example is yogurt and the only existing health claim, because it's actually approved using the codex definition. So we should have a codex definition for probiotic and postbiotic. And everybody knows that it takes 4 years to get something through in codex, but at least it's then acceptable for the regulatory authorities. The other point is that we see more and more non-QPS bacteria coming into the research world. This morning somebody was talking about inactivated Prevotella. Definitely a novel food, and it would be nice to have a consensus definition for that purpose also, because the assessment is quite different, and it requires safety assessment, and it requires, as Gabriel informed you, methods how to analyze the products. And if we want to see novel development, then consensus is required on that side as well.
All right, on that aspect of product development, perhaps Guus, can you share your insight on the industrial side of things?
Yeah, I would happily to do that. maybe first start with, for an industry perspective, this is actually a wonderful thing that we have now, this debate, because we're really talking about a leaf on the biotics tree that is, on the one hand, relatively new, the postbiotics. On the one hand, it may be something that is very old, maybe the oldest thing, and people have been using it for centuries without having a proper name for it. Actually, when I saw the paper, the ISAP position paper, of course, we were very happy that ISAP takes initiative and leadership in this. I was even equally, or maybe even more excited when I saw the discussion raised by Morelli and many other very experienced scientists. And actually, this was really how I imagined that science would be when I was young. This type of debate, and it's all very positive. Maybe now making it a bit practical from an industry perspective. Our experience as a company goes really with applying a process. Maybe I give this example right now. Applying a process of partial fermentation in the production process of an infant formula, with which we had very interesting results, very interesting experience. But actually, we were using this already for a long time, and we were not calling it postbiotics. Actually, having a name for it, it's actually very, very useful to have a debate, to also decide what is it and what are the specs we should look at, and how can we improve this process, or how can we continue development. It was already mentioned by Gabrielle. Probiotics, it's quite easy. You can just say, okay, do we have viable cells? Okay, then you know that your production goes well. If everything is dead, you're not making a useful product. Postbiotics is difficult because postbiotics is so many things. What we have seen, for instance, that if we have make tiny alterations in our fermentation process, changing conditions, it already can have a different outcome. So how do we as an industry know that we are, and we're not even talking about legislation and consumers, but how do we know if we are doing the right thing, if we are making the high quality product that we want to make? In order to do that, you need to kind of define what you're making and also know which component of this mixture of postbiotic components is actually the most relevant to achieve this health benefit. And I think this is quite we can really come back to the kind of the measuring or the advanced analytical science that you need to do that, because otherwise you have no idea when you're making your right postbiotics products, or whether it's you have to change something. So I think that the fact that it's so dependent on conditions, I think it's really difficult to know what are your criteria of product quality. And there actually I don't see a real kind of framework also in the current definition. To be honest, I also really don't know how we can solve this with a definition. I think it really matters from postbiotic concept to postbiotic concepts. So this is a long answer, and I'm not making it clear. I realize, but maybe
it's making it more difficult. Hold on, Gary. Can I first have a response from Lorenzo?
Well, thanks, Kun. I think that first, we really need something arriving from FAO Codex. I think it's time that the national government, there are only the national government that can do this, ask to FAO to convene an expert group in order to have these kind of things. Also because I'm thinking about the million of euros that the food industry has used for research in the probiotics area. And nowadays, in a lot of European countries, the term probiotic is banned, but it is used by the cosmetic companies, by the detergents company, and so on. So I think that I totally agree with Seppo. We need something from FAO and Codex. And concluding, I totally agree with the last comment. I really think postbiotic could provide to science, to consumer, to industry, a lot of very interesting improvement, but we need to know which are the components that we have to monitor. We have tried to do a lot of animal studies using exactly the same strain, derived from the same bioreactor, dividing into two the bioreactor, but the inactivation steps were different. The results were horribly different.
Okay, clear. Thank you. Gabriel.
I think this is the same problem or the same issue we have with probiotics. We all know that, for example, with LGG, I have no conflict of interest, but if you produce LGG with a different temperature and pH, the adhesion will be different and the effect will be different. So the control of the process is key. It's a key to produce something that is reproducible. And I'm not sure that we have to know exactly which is the molecule or the combination of molecule that produce the effect. That is the mechanism of action. And we don't know the mechanism of action of all the probiotics. And again, coming back to probiotics, we focus our attention in the viable cells. And if you get a probiotic product, you can see cell debris, you can find dead cells, you can find metabolites, and you're not paying attention. You just give the product and you have a health benefits. If you know
Gabriel, the dose response studies published have been done on viable cell counts. So you can see in the graph the viable cells and the health benefits.
Yes.
So what is known now?
Yes. But I think in those 1 year studies where they give a probiotic during 12 months and they give the same batch, do they know how many dead cells are accumulated in the product? And so that I think have to pay much more attention in non-viable microbes.
So we have some interest from the audience to participate in the discussion. Can I have the microphone to the front here?
I like the idea of throwing the microphone.
Yeah,
I never saw that.
Just throw it. It's a soft one.
It's quite fun.
Thank you. Wilbert Siebesma, Microbiome Solutions. I would like to ask especially to Gabi about fermented foods. You are publishing that a lot on your Instagram account. And if I think now about a glass of red wine or a pasteurized yogurt or now there are many you know much better than I, or maybe also a piece of bread. I think all of these contain fragments, maybe even a cup of coffee, contain fragments of bacteria. They have proven health benefits quite often, and they contain the metabolites that were produced during the fermentation. Would you say, also to make it easier for the consumer, don't know, are these all postbiotic products?
No, because depending for the postbiotic or a probiotic, you have to know exactly what strain you are using. And when you are using sauerkraut, you are using when you're using wine, when you are doing kimchi, microbes change from batch to batch. Maybe there is a core of microbes that are responsible for the effects, but they don't strictly qualify a postbiotic because you don't know what strain you are using. So maybe it's a putative postbiotic, potential postbiotic, but to be strict, you have to know the strain you are adding that gives reproducibility.
Okay, good. Thanks for that reflection.
So something that is measurable, then again, you know, the correct strain. I'd like to contribute to the discussion a little bit also when you mentioned LGG. I know that also Wilbert was involved in a study where part of the genome of LGG was kicked out by the bug, and that happened to encode for pili, which might have been the probiotic factor of LGG or not. We still don't know, but 70% of these cells in a commercial product had that genomic deletion of these pili. So genomic stability then also is an issue, and it may have been the industrial process that led to the deletion of that genomic island, right? We don't know, we can't figure that out. But again, that boils back to being able to measure what's going on in such a product. Seppo.
I think the important thing is that we should be able as scientists to define what to measure, because when we are not doing it, then the regulatory authorities are doing it. And we can see that we have three potential postbiotic products in the European Union: Clostridium botulicum, which was approved before the novel food regulation, and then Bacteroides xylani solvens, which is clearly in food, and EFSA has defined how long it needs to be pasteurized so that non-viability is proven. And then Akkermansia, you can read the details in the EFSA evaluations, but if we are not doing it as scientists, then the regulatory authority is doing the definitions.
We'll do it for us, okay.
And I think for quality control, you have to think in something feasible. Me as a regulatory agency, I will say, okay, for any LGG product, you have to go and measure the pili. That's impossible to make. Maybe it's difficult in Europe, in South American countries, that's completely impossible. So you have to use something, okay, let's be flexible, maybe see a few, it's okay, until we know how to go more in detail.
May I add a very new information about this, about the last comment by Gabriel. This could be true for food use, but 2 weeks ago, the first probiotic European pharma product, an Italian one, has been approved. And one of the requirement by the European Medical Agency was to perform the whole genome sequence twice a year on the master cell bank.
That's a good one.
That sounds like something that can be accomplished, yes.
Yeah.
Can I ask, because I'm still thinking about how do we know that we're making the right stuff when we're producing postbiotics. What I hear is I have the feeling that postbiotics basically by default is a journey, because the more you understand the mechanism of action of your postbiotics, the stronger basically the obligation becomes to crack it. And it eventually, something that starts as a postbiotic will always end up, I mean, ideally in molecular medicine. Is that something, or am I now too provocative? But I have the feeling that if we follow this line of thought, you basically, Gabriel, you also mentioned this, you end up with something holistic. You start to understand how it works. You measure then which component does the trick, and you start to measure the concentrations in your process, and at the end you will end up with one molecule, or maybe a few of them. Is that a kind of deterministic journey, or is that not the case with this definition?
Seppo is very eager to respond to that.
I was not actually responding directly to that, but by saying that ISAP also has had the idea that we should reach beyond Europe and North America, because we too often speak about European Union regulations or American regulations. Now, Gabriel is bringing us South America. We had Dr. Endo from Japan in our previous one, actually to also introduce the ideas that are present elsewhere, beyond Europe and beyond US.
That makes sense, yeah. Do you want to respond to Guus's provocative remark?
I think it's really interesting to know all this, to crack and to know, but I think it's something that it's really difficult, challenging to do, because sometimes, okay, I have this very small metabolite, I won't pay attention. Maybe it's just a small amount because it's enough to produce something. So it's really, and imagine how many metabolites fragment you can find there too. I think for the moment, okay, I will produce this. This is my process. I know how to manage the process, how to reproduce the process. And with this, I have this effect. And maybe there is a flexibility, because we know for probiotic, it happens. In many conditions, you change the dose and you don't see a huge change in the response. So there is like a kind of range that we don't know, flexibility, that you can move your process. Okay, I will ferment a little bit more, and still have, and again, the probiotic or the postbiotic effect is not quantitative. You won't say this postbiotic will reduce 13% the chances of blah, blah, blah. It will give you, promotes gut health. It's really a qualitative thing. You're not giving exact, what you won't say it enhanced IgA in 15%. You will never say that. So I think we have some flexibility that we don't know how big is it.
I'm very
If
curious
I can
what
add,
Morelli wants to say, yeah.
Yeah, if I can add, I really appreciate the fact that we must perform a worldwide observation, but just think about the class action in the United States against food products that have been marketed promising something on gut health, and then there was a class action. So we have to be extremely careful in using this kind of position, especially for the companies.
Okay, thank you very much. There's one more thing that I want to address where I saw, I heard more a discrepancy between what Lorenzo was saying and what Gabriel was saying, and that was Lorenzo's focus. I think if we go back to that word cloud, then one of the other words in there would be food. And Gabriel, you were talking about the potential to have inanimate pathogens that could still work on the immune system, right? So pathogens sometimes are present in food, not supposed to be there, right? But I think there's a discrepancy there. Do you propose to add those to food, and what would that mean to regulatory bodies to have these kind of postbiotics also?
No, of course, I don't expect them to be in food, but maybe we have a long history of bacterial lysate as respiratory booster. So they qualify as postbiotic. They are preparation of inanimate microbes that confer a health benefits, that is improve immunity. So it's like a supplement.
Okay, so but then already existing products is what you were referring to then?
In Argentina, they exist.
Yeah, okay, all right.
But they exist also in Italy, but they are classified as drug.
As drugs, right? Yes. But in Argentina, can they be food supplements or?
They are free. They don't. You don't need a prescription.
Yeah. So there's clearly differences in
yeah,
regulatory affairs,
yeah,
right?
yeah.
So, and there I think Seppo's remark about unifying all of those different regulatory bodies across the world would be very good to do. All right, I'm looking at the audience. Are there any urgent questions from the audience? Yes, we have one over there. Wilbert, can you throw the microphone? Careful.
Got it. I'm doing a PhD right now on postbiotics and the gut microbiome, so I've kind of studied a lot of your stuff. It's been very interesting, this discussion. I have a number of questions, but I'll just say one now. For the definition versus the regulation, do you have your opinion about this is the definition, but then when we go to regulation, we're going to have a list of the regulations that we're going to need to state by the supplier of the postbiotic, say how we've produced it, fermented it, what are the conditions, what the medium, how we inactivated it. So we say it is a postbiotic, but for each case we have to say how we went about producing it and how it's prepared and concentrations, or is it's a postbiotic of Plantarium and therefore you can just use it, call it postbiotic?
I don't know if I understood well the question. I participated in presenting dossier in Argentina to our regulatory body, and they ask a lot of detail how you prepare, and these are confidential. But of course, the regulatory body will want to know how you produce your product, either being a probiotic or postbiotic, and you have to give all the details that of course you won't publish because it's your information, but the regulatory agency for sure will ask for that.
If you then say for the companies that are using the research that you've done on a postbiotic of Plantarium, for example, can they then just say, oh, it's now like how probiotics, all the companies and all these labels say, oh, it's a probiotic of whatever it is, without actually doing the studies on it. So are they then required to use the same conditions that you or whoever published the work on that specific postbiotic to then call it a postbiotic, use the same conditions, or is it we've just fermented it and inactivated it and therefore it's postbiotic?
Do you want to answer from the industrial point of view? I think processes are really important, and we speak.
Absolutely. This one? Not sure. Okay. Yeah, I gave this example already. So how fermentation conditions can have a huge influence on the efficacy of your end product. So yeah, this is something you have to master, and maybe this kind of connects to the kind of almost kind of artisanal part of postbiotics when it's used in kind of traditional food. Yeah, maybe it's even in modern times still the case. You have to be extremely, how you say, capable of controlling the fermentation conditions and also understanding which conditions make the difference. So that by default requires a lot of study. Of course, maybe the one postbiotic is not as complex as the other. I think I can give also again an example from a concept my company has worked on, where we used in fact 2 organisms that together were involved in the process of fermentation, but clearly had very different roles in changing the pH, in making certain metabolites. And yeah, we've learned basically by empirically that it matters a lot when you add the organisms in the process, in which state you add them, because it relates to the kind of the viability or the activity. We all know, we're all microbiologists, we all know about how the growth curve of a microorganism, and it makes a lot of difference in which state the organism is added to the process. I'm just giving you some examples of basically how challenging it is and how much investigation and development it requires to make a postbiotic concept that achieves health benefit, especially when you talk about specialized nutrition for patients or infants. It's yeah, although it may be a bit old school and traditional, appeals to fermenting food, but actually it's a highly technical yeah, I think industry, especially if you want to control it and prove that it's efficacy. So does that give you some kind of answer?
That's good, thank you.
I think one could add to that also that if you really want to have novel food approval or a health claim approval, then you need to define your process step by step very carefully.
Okay, well, I'm going to cut the debate short. It's time for a networking break with coffee and posters. I'd like to thank all of the participants in the panel. Give them a great hand, please. And I think we can unify them. I think indeed we can have this starting definition, and then we can add to that. You know, I think discussion in science is a great thing, and I've already heard the speakers agreeing with one another on certain aspects, so that's good. So let's see where this goes in the future. Thank you very much. I'll see you after the break in 25 minutes.
Okay, I will go for an espresso.
Thanks, Lorenzo. Bye-bye.
I will go for an espresso, huh?
Yeah, we do.
Bye.